Yeah.
I feel like there's nobody here right now. I've had a great 20 seconds because I just started my thing
Oh,
like
yeah?
great 20
I
seconds.
gotta like subscribe. You should make this public. You really should.
And I
They
go
are?
live. Yeah, when you go live, they're already made public.
So after it goes live, it's like still on your account
Yeah.
as public. Can I have your?
Madison Brown, that's my name.
It's just Madison Brown.
Yeah.
Like nothing else. It's just Madison Space Brown.
I think so.
All right, I will look it up and subscribe because I need to review that.
See, I name the recordings. Sometimes they're correct, sometimes they're things changed in the middle before I actually knew what was going on.
Yeah, right. That's like when I name my nodes. Like I named this one the citric acid cycle, but it's not. It was pyruvate oxidation. So it's a completely different thing.
Dang, a minute and 20 seconds without anything really come out. How long will this thing go without anything actually happening?
Hopefully all class.
No, I need those notes.
Yeah, I need the notes too. I need them so bad.
And I realized our little thing, the test thing, it's on Halloween.
What time? Oh, next Wednesday, right?
Yeah.
Sweet. Oh no, not sweet.
Yeah.
An exam on Halloween.
I had a plan that I was gonna go to my old high school and go pass out like some baked goods I was gonna make.
Oh yeah?
But now I'm thinking, well,
You got an exam.
yeah, and I can't get back until like 1. But that's like almost everything.
Yeah, that's too bad. What do you
Yeah.
feel like in the morning?
Yeah. I need to figure out what I'm doing.
Maybe remake the exam. I don't know.
No, that won't be worth it.
Right. I think so too.
I even had a plan of dressing up all day, handing out some snacks.
Aw, that's too bad. You had a whole plan and everything.
Yeah, I bought a costume and everything for this.
Yeah. Yeah. I have a costume ready for Halloween too. It's gonna be awesome. I'm gonna walk around with my brothers. Like my little brother's gonna trick or treat. He's 9, so this is the first year he's decided to like dress up scary. You know,
Ooh, instead of
he's that
cute.
savvy. Right, exactly. Because before he would be like a superhero or something,
That would be awesome.
but now he's like scary.
I'm gonna be scurry.
He's so scary.
I'm scurry.
He's all into it. He's got like one of the masks that has like fake blood in it, you know?
Oh.
You pump it. And fake blood comes down.
I feel like every time, even if I tried to be scary, it never worked.
Yeah, I tried to be scary once and it just wouldn't go. So this year, me and my brother, my older brother, are dressing together as like 80s action stars.
Ooh.
Like the Terminator and like that kind of look, you know, like leather jackets.
That'd be pretty cool.
Yeah.
I remember one of the costumes I did. I went as Death. I think I was.
As death.
Yeah, I had this big old leather black cloak thing, and I just painted my face as a skull and stuff.
That's so awesome.
I won a costume contest,
Oh really?
although there was only 3 entries in there. I won, gosh darn it.
For my calculus class, if we dress up as a famous, I forget who it is, but it's like a famous woman who's like did something important in calculus. If we dress up like her, then we get extra credit on the exam.
What about the dudes in there?
They have to dress up as the woman too. So
Wait, is this in your class?
yeah, and she has to like wear a bonnet. I think she's wearing like a bonnet. Did you do that for calculus?
What?
If you dress up as I forget her name, but someone important in calculus, then you get extra credit on the exam.
I won't
Go,
miss in this
folks,
class.
let's get started.
You're such a good person.
Looking at the material we've got, I think we'll put the test off the next Friday.
I love that.
We got a lot of material to cover.
I'll send you that.
I'm
So
What's
can
your
go in the
phone
morning.
number? Here,
we'll
put
do
your
it
phone
next
number there.
Friday.
Well, I'm gonna probably come.
Thank you, Dr.
But since I don't have to study early in the morning, it's gonna be better.
Oh, yeah.
We started, folks, it's gonna be a tough test, you better listen. It's gonna be a make or break for a lot of people. We started talking off about cellular respiration as the way we extract energy from molecules. The primary method is from carbohydrates. And the primary molecule that is used, food molecule, is glucose. Some things you need to remember. Number one, remember the equation for oxidation. C6H12O6 plus O2 is going to yield CO2, water, and energy. Now we want energy. So, your body, remember, prefers to metabolize and to use carbohydrates for energy because of the clean waste products, carbon dioxide and water, are very compatible with your body. Plus, they are very easy to break down, so your body can get most of the energy that's in the carbohydrate out. And that is not the case with fats and proteins. Even though they do have more energy, we can't get it out. So, or it takes us a lot to break it down. Net gain of free energy that we get. So we start with the glucose molecule. We looked at the steps of glycolysis, and at the end, that glucose is turned into two molecules of pyruvate. During the process, by substrate phosphorylation, there will be 2 ATPs of free energy made in glycolysis. There will be 4 made total, but 2 of those have to fuel the reaction itself. So the only amount that is left for free energy use will be 2 ATP. From there, pyruvate must go through some changes and then go across the membrane. Yeah. Substrate, remember what oxidation is. Oxidation is to give up an electron. In substrate phosphorylation, instead of the substrate giving up the electron, it goes to an enzyme. And in that enzyme, whenever the bonds are broken, then it captures that energy and puts a phosphate on it. So substrate level phosphorylation is just breaking a bond in a substrate, using that energy to make ATP. That is done by an enzyme. In oxidative, the electrons and the protons are separated across a membrane, and that is the oxidative phosphorylation. Yep.
So you said that 2 ATP result from
2 net.
what's 2 net?
4 total are made. 2 has to fuel the process.
From glycolysis.
Next up, that pyruvate has, all of the glycolysis takes place in the cytoplasm. Pyruvate made in the cytoplasm now has to go across the mitochondria, into the mitochondrial membrane, and it does so by combining with another molecule, coenzyme A. This is pyruvate. Now, as pyruvate starts to go across, proteins, enzymes will help it go across. It loses a carbon dioxide. When that carbon dioxide is lost, it's just given off into the atmosphere. We have this molecule left, which is an acetyl group. What you see in red and black are acetyl groups. That's carried across the mitochondrial membrane by the proteins. Carbon dioxide is given off. It then combines with coenzyme A to make acetyl-CoA. This little group is the same as that group right there. The only difference is that coenzyme A has been added to it. The coenzyme A is basically a transporter. It's going to take it to the places in the cell where then it can start the citric acid cycle. In that process, we get NADH made. How many molecules do we say were made
Four.
of ATP total?
Four. But two you were used. Two
Well,
are.
one of them, right, but the four that are made, one is by substrate phosphorylation there. And then you've got three that can come out of the NADH. You can make three. So there's four.
So one's coming from substrate phosphorylation, and then three from.
So they're all coming basically from substrate phosphorylation in a way. But this NADH will move on to the system.
And that's only 3 molecules, you're saying?
It's just done inside the mitochondria there.
Okay.
Yeah. And we've got 2 molecules of pyruvate, so we're doubling things.
Right. So do we need to remember where about
No,
those 4 molecules?
Not with ATP.
Okay.
It is confusing, and it changes depending upon cells and things, so it's not a set standard way.
Okay.
Now, once we get acetyl-CoA attached to pyruvate, notice pyruvate starts out as a 3-carbon molecule. Glucose was a 6-carbon molecule. It was broken down into 2 3-carbon molecules. So one of these pyruvate attaches to coenzyme A, and in the process gives up a carbon dioxide where it loses 1 carbon. So now we have got the acetyl group here. It comes in to the citric acid cycle or Krebs cycle and combines with a chemical oxaloacetate. The acetyl, remember the CoA is given off. The acetyl group combines with oxaloacetate. When it does that, we have 1, 2, 3, 4, 5, 6 carbons again. So now we are to citrate or citric acid. Remember the difference between the acids or the ate forms is whether it's ionized or not. So this is citrate, or you could call it citric acid if you wish. Now what happens there? You don't see this, we do not have this on the board, but there are many, many enzymes that start to go to work on these. The result of those enzymatic action, water will be produced. What happens when we break that water, hydrogen off of citrate to make water? Energy is released, right? And it's going to be picked up by the NAD to become NADH. We have to keep this molecule balanced. Now, when we remove 2 hydrogens and 1 oxygen from citrate, we get what is called isocitrate.
Do you need to remove all of these?
Yep, you need to know they're there. You don't have to know the diagrams, but you do need to know the products.
Step 2 is citrate and isocitrate?
No, citrate becomes isocitrate
When it releases the
after it loses the water, because you're giving up 2 hydrogens and an oxygen. So that's not the same molecule anymore. We've taken 3 atoms away from it. So it becomes something else. It becomes isocitrate, and that, taking out that hydrogen and oxygen, releases energy that is picked up by the electron carrier. Then, one of those hydrogens go to the NAD, and there's another hydrogen here in the system still, proton. What do you have left? You've got an electron up here and the proton, right? We've separated those. We take away the electron, what do we got? Hydrogen proton. Next step, another enzyme will come in and removes another oxygen. When it removes, or another carbon dioxide, when it removes another carbon dioxide and oxygen, notice what we've got. 1, 2, 3, 4, 5 carbons now. So we've lost a carbon and an oxygen, 2 oxygens, so that's not the same molecule that we started with, isocitrate. Now it becomes this molecule. Carbon dioxide is given off, remember. Alpha ketoglutarate, which comes from alpha ketoglutamic acid. Yeah.
So it's not shown, but these are in each step there's an enzyme acting
There are
on
many enzymes, yes.
on it. Okay, so when it's showing in step 1, okay, so when it reacts with an enzyme, it becomes, I mean, when the, let me see here, when the acetyl acts with an enzyme, then it becomes
It
oxaloacetate?
combines with oxaloacetate to make citrate.
Okay, okay, and an enzyme, well, influences it there
Enzymes
also.
do it all.
Enzyme do it all, okay.
So
Thank
here
you.
we are at alpha ketoglutarate. Enzymes react, another carbon dioxide is given off. Notice here, coenzyme A comes back and combines with what is left over after we take away the carbon and the oxygen. And notice there are once again enzymes, the carriers, the electrons are added to the NAD to make NADH. NAD positive becomes NADH. With the addition of that electron, we've got another proton in the system. Now remember, these yellow things are what is important that comes out of the citric acid cycle. Those electron carriers with the electrons attached that will make energy, yes.
Well, the ATP I saw definitely that's energy.
ATP is the form that we want out of the electron transport. This is what makes that ATP. So, this is what's happening all the way through here. Succinyl-CoA attaches once again to be basically a placeholder. And then it will move through the system. Enzymes will work on it. Notice what happens. It kicks off that CoA because it was just there as a placeholder primarily. Now we've got another inorganic phosphate that comes into the system that will wind up making ATP. So it's going to take that energy that has been built up to make ATP. Not the carriers, but these protons and so forth.
So the protons, okay, so the protons are using the energy built up so far to make ATP, and that's where that inorganic phosphate's coming in. That's where the
There's energy
ATP
being made in 2 ways. A lot of the energy is going in to make phosphorylate NAD, it's NAD, or not phosphorylate, but to add an electron to NAD to make NADH. Doesn't use all of those hydrogens in the process. So some will build up and actually be used to make ATP. No. NAD, remember, it can hold enough electrons and energy to make 3 molecules of ATP. So there's not always enough to even make an NADH. So that is being turned in to ATP. After that phosphate comes in, remember what's going to happen to the phosphate? It's going to be added to the ADP to make ATP. It's not going to become attached to this. Whenever we have these things released, take this excess energy, then we can make an ATP.
Is that considered substrate of phosphorylation?
Yes.
Okay.
The only place where there is oxidative phosphorylation is in the last stage. Some people call the last stage just the electron transport system. There's actually 2 steps in the last stage, which will be electron transport system and a process called chemiosmosis. Electron transport system takes that energy and carries to a certain place. Chemiosmosis is then taking the energy and making ATP out of it. Here is FADH is made, or FADH2. Notice we got FAD, it can pick up a couple of hydrogens. And in the process, it can contain enough energy to make 2 ATP. Not enough here for an NADH, but it's enough to make FADH2. So look what we started with. Succinate. How many hydrogens? It's succinate.
4.
4. Now it gives up 2, so we've got 2 then in fumarate. 2 of the hydrogens went to FAD. We have fumarate left. Notice here, water comes into the system. What happens when we bring water into the system? We add 2 more hydrogens and an oxygen. We will then be able to get enough energy to make NADH. After we take malate and extract enough energy from it to make an NADH, that can make 3 more molecules of ATP. Look what's left. Oxaloacetate. So the end of the Krebs cycle, after we start pulling these hydrogens and carbon dioxides off, remember, if we totally metabolize a glucose molecule, what do we have left? Carbon dioxide and water.
Oh.
So that's what's being pulled off of these. Should be none. None make energy. They change it and release energy. They do not make it. Sun is energy. We're not making it in any of these cycles. We're just releasing it. So keep that in mind. Any questions on that right there? That's what you need to know. You don't need to know where the sun comes off, where the hydrogen is. Know the products and know where energy comes from. Well, the first step is going to make citrate, yes. You will have acetyl-CoA come in and combine with oxaloic acid, and then the first step starts, that's citrate. Not necessarily. The product of the first step is citrate. And the number 8 is oxaloacetate, right? Yeah.
Okay, so saying that 1, 2, 3, 4, 5, 6, 7, 8. Okay, so the first one's citrated, and then it ends at oxaloacetate. But when it combines to make citrate, the CoA was already taken off, right? So it's only acetyl and oxaloacetate that combines to make citrate, right?
It's the acyl group, the acetyl group, and oxaloacetate together. The acetyl group is 2 carbon, oxaloacetate, 4 carbon. They come together to make the 6 carbon citrate or citric acid.
So the NADH and the FADH, they're carrying energy basically,
Electrons.
electrons, which would be energy. I mean, they're
Well, that's the way energy, the form of energy is in electrons.
and NADH carries more than FADH because you said that one NADH molecule can
Make
yield
3.
3 ATP.
FADs can make 2.
Make 2. Okay. So if we have, so this is considered like, in step number 8, we'll say. In step number 8, that's one NADH molecule, and it can make 3 ATPs?
Correct.
Okay, so that's what we have, basically, 3 ATPs?
If you don't have them yet, you've got enough energy to make 3 ATPs.
Okay.
It's still not ATP until that ADP is phosphorylated, and the reason there's a difference, that ATP is what's going to move it to the cell to release it in other parts. It's not in that form yet. Energy is energy, but what molecule can release it?
That's ATP,
That's
Kieran. And so basically, that's why you don't necessarily get 3 molecules out, is it? Because you have to put energy into phosphorylation? Is that what?
No. You have to put energy into the citric acid cycle, glycolysis to get it started.
Okay.
And the whole point of the electron transport system and oxidative phosphorylation is we don't put any energy into it. What you do, you separate. Remember, folks, you've got 2 magnets, positive end and a negative end, they want to come together. That desire, that physical attraction of them coming together creates a force. That force is what you use then to put phosphate onto ADP and then can release that energy, the force it took.
Oh, okay.
So it's not a flow of electrons. It is the attraction of protons to the electron that causes the force that makes ATP.
So you're not actually putting into, you're not actually putting energy into it because it's a natural force, right? So it's almost like diffusion. It happens naturally. It's not actively.
Some, and you'll get to see this when we talk about photosynthesis, some of that energy is just the process. There is enzymes and things like that working in there. So yes, there is a certain amount of input good from one standpoint, but it is not energy such. That's what comes in at the first that starts off with this and gets it going.
Okay, I was.
Remember, when we put it in at first, we're breaking things apart. Now they're not fitting the octet rule. They always got that force wanting to, and that's, we keep separating them apart and pulling things off, so they never get there until the end.
Okay. Thank you.
Okay. Remember, the citric acid cycle is, for all practical purposes, its point is to create, not create, but to release energy to create the electron carriers. So we've got 1, 2, 3 electron carriers. There's 9 molecules of ATP. Here's 2 more. That's 11. We've got ATP made right here. That's 12. We do this twice with 2 molecules. So now we're up to 24, and there's 4 made in here, so we get, that's where they're coming up with the 34 in. Yeah,
Did you say it was to create energy carriers to release energy?
correct. We can create energy carriers. We can't create energy. The energy carriers will then release the energy that they're carrying. It's like a battery. If the battery's charged, we get electron flow. If it doesn't, we don't. Yeah.
So the NADH is the energy or the electron carrier?
Yep. Remember what I said earlier. Think about it as if those electrons were berries on a blackberry bush. We're changing that shape of the bush so we can start to pull off those electrons. Those electrons are then picked up by the NAD and FAD and then taken to another enzyme at the end of the electron transport system that's going to use that to make ATP, which is what we're getting ready to go to. So we said, most important product out of out of glycolysis is pyruvate. The most important product to come out of the citric acid cycle is not energy, it's these carriers. They are not energy that can be released as free energy yet. the most important product to come out of stage 3 will be ATP. So let's take a look at that now. Let's clean up a little bit here. Notice it says the citric acid cycle has 8 steps, each catalyzed by an enzyme. So once again, we're not showing you the enzymes. You do not have to remember them. Just know they're there. The acetyl group of the acetyl-CoA joins the cycle by combining with oxaloacetate, making citrate. All the next 7 steps do is to decompose citrate back to oxaloacetate, releasing the energy from what? That acetyl group, right? Oxaloacetate and acetyl group combine to make citrate. Oxaloacetate is going to be regenerated, so the only thing we're getting pulling the energy from is just that acetyl group. Actually, from the old glucose molecule if you go through glycolysis. But in the citric acid cycle, it's only pulling electrons from that acetyl group. What happens to that acetyl group? It is converted to water and carbon dioxide. And then the only thing we have left is the oxaloacetate. It's actually a neat little system. If you guys go through and do anything in biology, if you go into a health field like medicine, pharmacy, you'll see this 6 or 7 times in other classes, and it's better to learn it now, I promise you. Because they're going to go into it much deeper than we are. This sounds like we're going into it deep. We're giving you an introduction. You can actually teach full courses. You can get a PhD in the Krebs cycle. So we're not going into it nearly that deep, and there's no reason to go into it that deep at this level.
Well, what about the fact that the law of thermodynamics may actually define what happens to your spirit after death? Because it's just electromagnetic energy, and since it can't be created nor destroyed, I mean, think about it. You're just going.
I
It's
think
quite
that's a whole different
possible.
thing.
If you could be out far enough in space, and if you had a device that could collect the sound waves and amplify them, you could hear Abraham Lincoln giving the Gettysburg Address. The waves are there, but they've been torn apart and changed, but the energy is still there.
Yeah, I mean, that does kind of make perfect sense when you come to think of it. I mean, it's more philosophical, but
Well, as it says, it cannot be created nor destroyed. It can only change the form. NADH, FADH, remember, produced by the cycle, relay electrons extracted from the food to the electron transport chain, which starts stage 3. Stage 1, we go to pyruvate. Stage 2, we tear apart that acetyl group to make the electron carriers. Now the electron carriers will be taken to electron transport. This just shows these being made. It doesn't tell you what's happening to them, where they're going. That's what we're going to do now.
Dr. Preston. So I know that NAD plus comes in and it takes up the electrons, which gets rid of its positive charge and it becomes negative, so it becomes NADH, that proton's still present on the outside, so they're separated, right? But the NAD, where does the NAD come from again?
It is an enzyme, basically, or there you go. It's a vitamin type nicotine adenamide dinucleotide. Is the name of it. It's in the system. It's always there.
Oh.
It's just like ADP. It's always in there. And once ATP gives up, a phosphate becomes ADP. Once NADH gives up the electron, it becomes NAD phosphate again.
Okay.
So it's always regenerating.
So you said it's an enzyme.
It's not actually an enzyme. I meant to say that it's like a vitamin
Okay.
or coenzyme.
But it just basically acts like a transporter, right?
It's a transporter, and it's always in system and regenerated by the system. Now follow with this. We're at the last stage The last stage will look to be extremely complicated, and it's really not. It's kind of neat. There's mechanics that start to work in this system. Now think about that. Could you have an engine just made up of proteins? Yes, you can. I'm going to show you one.
I thought you said you would die if you lived straight off proteins.
We're not living off. I said to make an engine.
Engine, okay.
If you take proteins that are certain shapes together and start putting them together, you can actually make a structure. Think of like if you looked at the cylinder of a gun where the ammunition goes into it. And you got those long chambers all the way around that cylinder, and down the middle there's an opening. Okay, if we've got hydrogen that can be pulled down that opening, it can make those other things turn because it's got electrons down below wanting to come together, so that force actually makes that turn. That's what happens here. And that's what it looks like, as we will see. Notice this is all very important too. Most of the energy is going to be in the form of NADH and FADH2 that we get out of food. Do we get all of the energy out? No. We still make carbon dioxide and water, and it takes energy for those to exist. So we don't get it all, but we get a lot. These two electron carriers donate the electrons to the electron transport chain, which powers ATP synthesis via oxidative phosphorylation. So now, we're leaving the Krebs cycle, and we're jumping into the electron transport chain. So it's important to see what it is. The electron transport chain is in the inner membrane of the mitochondria. Okay, now the citric acid cycle takes place in the middle, the cytoplasm, basically the mitochondria. Now, these electron carriers are going to move to the walls. Notice, citric acid cycle taking place here in the middle of the mitochondria. These are the inner walls that come in. That is where the electron transport chain will take place. And the reason you have all of these little things, projections inside, which are called crista, they are there to increase the surface area so we can make a lot of energy. If we just went around the outer membrane, it would not have nearly as much surface as we do right here. So that's what that's for. Now, those electrons made in the citric acid cycle are going to move to that inner membrane, the crista of the mitochondria. The electron transport chain is going to be made up of a series of proteins. You do not need to learn each and every one of these proteins in this course, but you need to know the groups, basically, that they're in. Notice these proteins come together to make multi-protein complexes. The carriers alternate reduced and oxidized, alternate reduced and oxidized states as they accept and donate electrons. So NAD positive is going to be reduced because it picks up an electron, then it's going to go to the electron transport chain. It's going to become a reducer itself. It's going to give up its electron, become oxidized, and then that electron will be on the membrane. That's what it means when it says it will alternate between reduction and oxidation. Notice electrons drop in free energy as they go down the chain. So, as we talked about with the law of thermodynamics and the idea of chaos, no system is 100% efficient. Some is going to be lost, and it's telling you here, every time they go down one of these proteins, they're losing a little bit of the energy that they carry. That goes off and makes what? Heat, primarily, in the system. Notice it says eventually, as these electrons carry that energy and they're starting to drop off, in the end, what's going to happen? They drop in energy as they go down the chain and are finally passed to oxygen, forming water. Because they will pull their hydrogen proton back to them again. Now we've got water. This shows you the multi-protein complexes. As I said, you do not need to memorize those. I don't see any point in that at all. Note that these will contain some proteins. They will be called cytochrome type proteins because of what they have in them. And there will be iron involved in this. Now look what happens here. This is the electron transport chain. We got NAD that was made in the citric acid cycle. Comes in, drops a couple of electrons to make NAD positive. FADH drops the electron to make NAD. Whenever it drops it, the NAD leaves, goes back to pick up more electron. Now this electron is going to go down this chain, pass from one protein to another. Like if we had a long line in here and one person at the end had a basketball and they just kept handing that ball to the other until they got to the end. That's what's happening. You can't see the electron here, but it's telling you it's coming down. At the end, it will come out on the other side. Now what did that do for us? It done one thing.
It prevents it from being an explosive reaction?
That does prevent it from being the explosive reaction. But what have we changed about that electron? Only one thing. Its location. And we will see it will be moved to one side of the membrane on the inside of the mitochondrial membrane, whereas the protons go to the outside. And then Adriana just said, look at the bottom. It breaks large free energy drop from food to oxygen. Does that in small groups so you don't get too much energy released all at once. Now, this is the second part of oxidative phosphorylation, called chemiosmosis. Oxidative phosphorylation has two parts: the electron transport system, which will position the electron in the correct place and do so without releasing all of its energy at once, no explosive reaction. Chemiosmosis will then use the energy that's in that electron to make ATP through oxidative phosphorylation. Now here, this is telling you what's happening, so read this carefully to get your understanding. Electron transfer in the electron transport chain causes proteins to pump hydrogen, protons, now, from the mitochondrial matrix to the intermembrane space. The protons then move back across the membrane, passing through a proton, ATP synthase. So what we've got, and what it's trying to tell you here, what's happening. We've got a membrane. That electron transport chain is going to put the electrons down here. The hydrogens are going to be pumped on the other side of the membrane. This arrangement right here arises because of the electron transport chain. Now, there will be a big enzyme in which that hydrogen is going to be pulled down through. The force will make ATP, be used to phosphorylate ADP. What comes out the bottom? That hydrogen proton. It's not changed, it's just the pull from these electrons. These electrons will be picked up by oxygen, and when we look at both of them, we get water. And you'll see a better drawing of that, but that's what's coming. The enzyme that the hydrogen is pulled through is called ATP synthase. ASE, an enzyme. ATP synthase uses the exergonic flow. What does exergonic mean? Gives off energy, the flow of that hydrogen moving to drive phosphorylation of ATP. We have got positive charges on one side of the membrane, negatives on the other. There is only one way that they can reunite, and that is to come through that enzyme, ATP synthase. The flow of that proton down through that enzyme causes a force, which then makes ATP. Once that flow is over and the hydrogen's through, it will reconnect with its electrons through oxygen, H2O, to make water. That is chemiosmosis, the use of energy in a hydrogen gradient to drive cellular work. The hydrogen gradient is separating the protons and the electrons. And there is ATP synthase, an example. That's the one I was telling you about. Looks like an engine. Actually looks like a generator.
Does.
Notice what happens. This is called the rotor. That part can actually turn. Notice it's showing you it's turning. Down here, we have a negative environment. We've got positive up there. The positive is pulled down through that, causing it to turn. Then after it turns, one will be dropped off. That's the hydrogen that's going to go to make water. That force turns all of these, and that force then is used to add the inorganic phosphate to ADP to make ATP. This is the membrane that you're seeing. And that's on the other side of the membrane. So that's all that's happening.
Did you say something about negative charge?
Negatives are down here.
Okay.
That's why the hydrogen wants to come back. But the only way it can come back is through that enzyme. That is ATP synthase right there.
Okay.
And they even give the parts of it the same number of parts rather that they do for a generator. The rotor, stator. You don't have to know those parts. I want you to know what happens there. Hydrogen is pulled through ATP synthase. The force it creates will then be used to phosphorylate ADP. Once the hydrogen comes out of the enzyme, it combines back with its electron that's attached to oxygen now to make water.
Is the force created or is it generated?
The force can be created, the energy's not.
Okay.
The energy's what makes the force.
You said the force is used to phosphorylate ATP?
ADP to make ATP. This is everything in that third stage in one little graph for you to look at. This is the electron transport chain. Notice what's happening. The NAD puts its electrons in these systems. The yellow line represents the electron. It carries through and then releases it on the inside. What is going to be the acceptor of that electron? Oxygen, remember. Hydrogen is pumped through these big complexes that we talked about to the other side of the membrane. H positive represents the hydrogen proton. It can then be pulled down through ATP synthase. That was the one picture we just saw with the rotor and the stators. As it is pulled down, it uses the force, the energy from that force, to phosphorylate ADP to make ATP. Then the hydrogen is released. And what happens to the hydrogen? It is what will make water. It will combine with oxygen to make water. Notice it says 2 hydrogen and 1/2 of an oxygen molecule because it's O2. We only need 1. So that other oxygen will be used to make more water. Now this is where, when you eat food, you're actually making a few water molecules.
That's the thing I asked you earlier.
And remember this, which is very important. What happens if we do not have oxygen here to accept these protons to make water? Water can then move across that membrane. If we do not have oxygen, the hydrogens will come through, they will build up and have no place to go to. Everything then, whenever this gets full, no more hydrogen can come in, everything stops. Energy production stops. The only reason we need to breathe oxygen is so in this step right here, it will be available to combine with the hydrogens, make water, and move out of the cell, out of the mitochondria. If it didn't, hydrogens would clog up in here, and it'd be like putting your hand over a water pipe. Even though the flow was trying, can't go nowhere. This has to move these hydrogens out of this mitochondria by turning into water. And that's it. 2 stages. Electron transport, chemiosmosis, they make the 3rd stage, oxidative phosphorylation. We'll see you Monday.